Hello everyone, this is Javaria and today I'm going to present a research article from Nature Cardiovascular Research which is entitled as the angiopoietin receptor Tie2 is atheroprotective in arterial endothelium. Let's have a brief look at the background of the study. So the main focus of the study is Tie2 receptor, which is a tyrosine kinase receptor. Before describing how Tie2 receptor is actually contributing to either the progression or regression of atherosclerosis or atherosclerotic plaque formation, let's discuss the role of angiopoietin type pathway in vascular biology. In a healthy vessel, a level of angiopoietin 1 is expressed at a high level as compared to angiopoietin 2. So when angiopoietin 1 bind with the Tie2 receptor, it actually initiates intracellular signaling, which ultimately lead to the vascular stabilization or vascular stability. But in a pathological condition or vascular remodeling, what happens is the level of angiopoietin 2 is expressed at higher level. When angiopoietin 2 binds with the Tie2 receptor, it actually negatively regulates the it actually negatively regulates the Ang1 Tie2 signaling access by promoting inflammation, vascular leakage, or leukocyte extravasation, which ultimately lead to the vascular destabilization. So, Tie2 receptor is predominantly expressed at arterial endothelial cells. So in this study, researchers actually tried to explore the function of Tie2 receptor by deleting the Tie2 receptor from arterial endothelial cells by using the Cree-Loxp system in order to visualize their function. And so after Tie2 deletion, what happens is it actually leads to the enhanced endothelial adhesion molecule expression and accumulation of immune cells and the expression of inflammation-related genes and FOXO1 nuclear localization. And all these processes ultimately lead to the increased atherosclerosis. So experimental design. And researchers actually took the advantage of multiple jivas studies. They did some sort of meta-analysis of multiple jivas to investigate the association of Tie2 gene with the coronary atherosclerosis, and they identified the top associated intronic variant and associated with the expression of Tie2 in tibial arteries. Then they used a technique which is CRISPR-mediated deletion and inhibition of the enhancer region, which ultimately led to the significant decrease in the Tie2 messenger RNA expression, which is showing that this variant is actually contributing to the change in the expression of the Tie2 receptor. So they actually investigated the effect of Tie2 deletion in the arteries in mice subjected to hypercholesterolemia by the systemic expression of the activated form of proprotein PCSK9. And actually they fed the mice with the Western diet, which is a fat-rich diet. And then to delete the Tie2 gene in the aortic arterial endothelial cells, they cross the mice carrying the Tie2 gene, in which the exon 1 is actually flanked by the LOXP sites. In this study, this mice is actually referred to as like the wild type. They actually cross this wild type with the BMX Cree ER2 deleter mice to obtain a mice in which the deletion of gene could be controlled temporally as well as spatially. So first result, here we can see that exon 1 is actually flanked by the LOXP site. This is a detailed schematic representation of the gene deletions in which the LOXP site is flanked by the exon 1. These LOXP sites are actually recognized by the Cree recombinase, and then they initiate the recombination. And then after the recognition, this exon 1 is actually flocks out. And again, this is in this experimental setup, what they did is actually they induced the deletion of gene at the 10th week by intravenous injection of the tamoxifen. And after what they did is they actually waited for 12 weeks for tamoxifen washout, and then at the 22nd week they gave Western diet to the mice, and also they get like they increase the expression of PCSK9 by using associated viral vectors by giving the activated form of PCSK9, and then they check the body weight and blood collection at all these weeks. In the figure C, the aortas of a wild type Tie2 and the Tie2 deleted it mice are stained with the Sudan dye, where we can clearly see that the atherosclerotic lesion formation is enhanced in the Tie2 deleted mice, is especially in the regions of thoracic aorta and abdominal aorta. This is the quantified result, which is clearly depicting that overall atherosclerotic lesion formation is enhanced in the Tie2 deleted mice as compared to the Tie2 wild type. But the more prominent obvious difference is at the regions of thoracic aorta and the abdominal aorta. All these results are clearly suggesting that arterial Tie2 deletion promotes atheroma formation. And in this, they actually took this, they did a single cell RNA sequence analysis of the cells taken from the atheromatous aortas from the Tie2 wild type and the Tie2 from the Tie2 deleted mice, and then they observed that the proportion of this is the UMAP plot, which is showing that the proportion of immune cells was higher in the Tie2 deleted mice as compared to the Tie2 wild type. But the difference is more obvious in the thoracic and abdominal aorta. These are the regions which are shown with the arrow. And this is again the quantified results, which is showing that the immune clusters the percentage of immune clusters is increased in the Tie2 deleted mice as compared to the Tie2 wild type, but the result is more obvious in the thoracic plus abdominal aorta. And in this result, this is a quantified result suggesting that there is increased immune cell infiltration in those mice in which the Tie2 receptor is actually deleted. We can see that CD68 markers or macrophages are actually increased in these mice where the Tie2 is actually deleted. This is aortas from the Tie2 wild type and the Tie2 deleted mice were stained with the Sudan dye, where in this state the mice were subjected to the Western treatment or PCSK9 overexpression for like 4 weeks, and where they observed that the formation of atherosclerotic lesions at the aortic arch. We can clearly see that in the Tie2 deleted mice there is increased atherosclerotic lesion formation, but the result is more obvious in the aortic arch as compared to the thoracic and abdominal aorta. In the same way here, they checked the increase in the inflammatory cells in the aortic arch, where we can see that there is increased immune cells in those mice where Tie2 is deleted as compared to the wild type. And this is the experimental setup where they did like the PCSK9 treatment and the Western diet at the 3rd week, and then they intravenously injected clodronate to deplete the macrophages, and this microspore is used to visualize the recruitment of the circulating monocytes in a real time in a intravital microscopy. And then they gave like anti-CD11B at the 13th or 14th week. To visualize the adherence of myeloid cells to the arterial wall. Here we can see that there is increase in the myeloid cell adhesion to the aortic to the external bifurcation area of the aorta in the mice where the Tie2 is actually deleted, and this is the quantified data, which is again suggesting the same thing, that in this area where the Tie2 is actually deleted, there is more adherence of the myeloid cells as compared to the wild type. And these all results are suggesting that Tie2 deletion increases the inflammatory cells in atherosclerotic lesions. Um, these are again these are the results where they give like the PCSK9 overexpression treatment and the PCSK9 overexpression treatment, and fed the mice with the Western diet for 8 weeks, and then this is the immunofluorescence assay, immunofluorescence staining results, where we can clearly see that in Tie2 deleted mice there is increased in adhesion molecule as compared to the Tie2 wild type. This is again the quantified result, which is suggesting that in Tie2 deleted mice, as there is increase in adhesion molecules. Again, these are the genes VCAM1, GLU, and FBLN5, which are involved in the inflammatory pathways and vascular remodeling. These all genes, expression of these all genes are enhanced in the Tie2 deleted mice, like in the aortic arch, as well as in the thoracic plus the abdominal aorta. And again, this is another clue gene in which also the expression is enhanced in the Tie2 deleted aortic arch, and as well as the thoracic plus abdominal aorta. Here we can see that there is more of expression is more enhanced in the thoracic and abdominal aorta as compared to the aortic arch. Again, they visualize the results after giving the PCSK9 overexpression treatment and the Western diet it after 20 weeks, where we can clearly see that in this immunofluorescence staining image that in the Tie2 deleted mice there is enhanced expression of adhesion molecule. These are again the quantified results, which are suggesting that the adhesion expression of adhesion molecule is enhanced in the Tie2 deleted mice. In the aortic arch as well as the thoracic plus abdominal aorta. Okay, here what they did, they actually gave Tie2 wild type normal Tie2 wild type were given normal diet as well as the Western diet, and they induced they enhanced the expression of PCSK9. Then they compared the cluster of immune cells, where you can clearly see that those mice which were given the Western diet and the enhanced PCSK9 expression, there is more immune cell clusters as compared with the normal diet. And in this, they compared the MFAB fibroblast under normal diet and Tie2 plus fibroblast under normal diet, where we can see that these all genes are expression or transcripts of these genes are enhanced in Tie2 fibroblast. And in this, they actually compared Tie2 plus fibroblast under normal diet with the Western diet, where we can see that those fibroblasts that were given with the Western diet, there is enhanced expression of interleukin-6, CCL5, C3, and many other genes which are involved in the inflammatory pathways. And in the figure C, what they are, this is the heat map, which is in which what they did, after isolating those fibroblasts, Tie2 plus fibroblasts, they actually given some fibroblast with they transduce some fibroblast with the control vector, scramble control vector, and some were given with short hairpin Tie2 lentiviral vectors, or it's in order to induce RNA interference, where we can see that the expression of these all genes are enhanced in those Tie2 plus fibroblasts in which the short hairpin Tie2 plus lentiviral vector is transduced. In these genes CCL7, CCL5, and C3 and SOX-S1 and APOD interleukin 6. These genes are mainly involved in the in enhancing the inflammatory pathway. These all genes are upregulated, and since these upregulated genes are involved in these pathways, and these pathways are also upregulated. Here we can see that in the chemokine CCL7, CCL8 genes, these genes are involved in these pathways, and these pathways are also upregulated. Here we can see like C3 and some other genes which are involved in these pathways. So these pathways are also upregulated, and in the same way, these pathways are also upregulated. In this stage of the interleukin 6 expression, where we can see that as compared to the control vector, there is enhanced expression of interleukin 6 in those fibroblasts, in which they actually transduced those fibroblasts with the short hair Tie2 lentiviral vectors, which is clearly suggesting that Tie2 actually restricts inflammatory signaling in the Tie2 plus fibroblast. It means that Tie2 fibroblast has some sort of immunomodulatory role. So here, again, they actually at this moment, they actually deleted Tie1 as well as Tie2. Then they actually stained the aortas of Tie1, Tie2 wild type, and Tie1, Tie2 deleted mice with the Sudan dye, and where we can clearly see that Tie1, Tie2 deleted mice exhibited more atherosclerotic plaque formation, especially in the thoracic and abdominal aorta. These are the quantified results, which are again suggesting the same thing, that there is a more obvious in the thoracic and the abdominal aorta. And this is here we can see that there is increased inflammatory cell infiltration, and after the Tie1 and Tie2 deletion. After the Tie1 and Tie2 deletion, and the effect is more prominent, and in the like abdominal aorta. Again, this is the this is the immunofluorescence staining, where we can see that there are more adhesion molecules in the Tie1 and Tie2 deleted mice, and this is the quantified result, which is again suggesting that there is increased expression of adhesion molecules like the VCAM and PECAM in the aortic endothelium. So the results are suggesting that Tie2 function actually dominates over the Tie1 in the atherosclerosis development. So this is again the immunofluorescence staining image of where we can see that after Tie2 deletion, there is increased FOXO1 and nuclear localization, and FOXO1 nuclear localization after the Tie2 deletion in the thoracic aorta, where we can see this is shown with the like red dots, and the DEPI is used for like nuclear staining or the background. As compared to the wild type, we can see that there is more like nuclear localization of the FOXO1. This is the quantified data, which is again suggesting the same thing, that there is enhanced FOXO1 nuclear localization. So results is clearly suggesting that Tie2 deletion promotes nuclear localization of FOXO1. This is the schematic summary of schematic showing a summary of the main results. GWAS studies showed that the association of TEK, which is a gene involved in the expression of Tie2, so its association with atherosclerosis, and then they developed the atherosclerotic model by giving the PCSK9 overexpression, the Western diet, and this is like control where the Tie2 is not deleted, and this is where the Tie2 is deleted, or either Tie1 and Tie2 both are deleted. Here we can see that when Tie2 receptor is present, it initiates intracellular signaling, and then what is happening here is FOXO1 is actually localized in the cytoplasm, which is ultimately destined to like proteasomal degradation, and adhesion molecule expression of adhesion molecule is inhibited here, while when Tie2 Tie1 is deleted, FOXO1 is actually localized to the nucleus, where it is binding to the VCAM promoter, and ultimately initiating its transcription or further expression here, which is ultimately leading to the leukocyte recruitment, plaque progression, and leukocyte adhesion molecule expression. So conclusion. So variation of arterial Tie2 expression is associated with the development of coronary atherosclerosis, and genetic deletion of Tie2 alone or in combination with Tie1 in arterial endothelial cells in mice actually promotes atherosclerosis. Thank you everyone for staying engaged. Looking forward to seeing you at the discussion session.